of these teams, this work would have been impossible. So all of these people should be fully commended for their amazing work. So a quick layout of what I'll be talking about today. So first of all, I'll give an introduction to the planktonic foraminifera and why I use them, why they're useful for studies such as this, and then a quick review of the Triton dataset, the methodology, why it was created, and why it's useful to studies such as this. And then using Triton for network applications in both the whole Cenozoic and at higher resolution in just the late Cenozoic. It's frozen for me, 2 seconds. Okay, so planktonic foraminifera, why do I study them? Why does anyone study them? Well, they have the best species level fossil record of any known group to paleontology. They have a very highly refined taxonomy, which has been worked on for decades by brilliant researchers and taxonomists. In general, they're being single-celled organisms on about the size of a grain of sand. They're highly abundant and generally cosmopolitan throughout the world's oceans. And because their skeletons are made of calcium carbonate, they extract the isotopic signal of oxygen and carbon from the water column, which stores a biogeochemical fingerprint of the water chemistry in their tests. And of course, their entire ontogeny is preserved within their skeletons as well. And using such things as the geochemical signal of their tests and just their external morphology, we can assign them to distinct ecological niches and morphological groups. So a quick recap on why and how we can use their geochemistry to determine what the ecology of not only extant but extinct species was. This is a profile of the water column, and with water depth on the y-axis and isotope ratios on the x-axis. And as we move up through the water column, we have a higher enrichment of delta 13C, and this is due to photosynthetic organisms preferentially extracting carbon-12 from the photic zone. And as you move up through the photic zone, more and more carbon-12 is extracted. So a higher enrichment of delta 13C is within the water column. And if you look on the right on panel B, also in planktonic foraminifera, when there are species which house photosymbiotic algae, which is illustrated on the left by an image by Richard Bisley. There's little yellow kind of circles around those beautiful little foraminifera there. And when they house these photosynthetic algae, they actually create a microcosm around their shell, which further enriches them in delta 13C. So we can tell the difference between asymbiotic and symbiotic species up in the photic zone. So also within the water column, as we move downwards, we have a higher enrichment of delta 18O, and this is due to the temperature-dependent fractionation of oxygen. So if we combine both these isotope signals together, here we have a nice G ruber, which lives in the top and is symbiotic, and a H scitula down at the bottom, which lives in the subthermocline. We can actually look at extinct and extant species and find out where they lived in the water column at the time. So here's a figure from a paper that I've recently submitted, which illustrates this a little bit more. So the delta 18O system is on the y-axis. Moving downwards, we have higher enrichment of delta 18O, and we move down, and there's our subthermocline forms at the bottom. There's our scitula, and on the x-axis we have our delta 13C. Moving to the right, we have further enrichment of symbiont activity and higher in the water column. So as you can see from our orange neoglobal Quadrina incompta, which lives in the subsurface, asymbiotic, and then our ruber in red is a symbiotic species. We have a higher enrichment of delta 13C. And using this isotope system, we can assign, Tracy Hayes and others assigned the species in the Cenozoic to different ecogroups. These include in the surface mixed layer symbiotic and asymbiotic forms, represented in red and orange, and these colors will be consistent throughout the talk. And then moving down in the thermocline, we have in cyan, that's a Globigerinola, In the subthermocline, Clavitorella, so deep in the water column with a stronger enrichment of delta 18O, and also species which tend to occupy regions of high productivity on continental margins and such. And then finally, our last ecogroup is our high latitude forms, such as Neoglobicodrina pachyderma, which dominates the modern day polar forms. So not only our 6 ecogroups we have, which we can define by our geochemical signal, we can also look at the external morphology of the planktonic foraminifera in the Cenozoic and assign them to 19 different morphogroups. And some of the morphological characters and traits we know do have a functional purpose, but some of them is still elusive to us. So despite them being assigned to morphogroups, we may not know the exact function of certain aspects of the external character of a test. So now, after the introduction of how planktonic foraminifera are useful in a study like this, I'll just give a quick run through of Triton again. So this was published last year, and we finally have a logo, which I created. 2 seconds, I'm gonna put a pointer. We finally have a logo, which I created a couple months ago. So that's the new Triton logo, which has the Triton's trident and a orbulina within the O, which is which I'm very proud of, obviously. And so the aims and objectives of creating Triton was to assess the completeness of previous marine micropaleontological data sets such as Neptune, and then create a new data set based solely on the Cenozoic planktonic foraminifera to help fill in a number of my PhD objectives over the last few years, and also, of course, to benefit for the overarching scientific community, as all the previous data sets have been fantastic resources for science as a whole. So a quick background on how most of this data is formed. It's mainly through ocean drilling, which was the advent of which was the Challenger expedition, and it's been through a number of different iterations over the last 50 to 60 years, and culminating now in, not culminating, but where it's now present is the International Ocean Discovery Program. And ocean drilling, how sediment is recovered. We have a number of different methodologies to recover those sediments full of microfossils. The first would be the very famous Joynes Resolution, which is a riserless drilling vessel. And then we have the Chikyu, which primarily operates offshore Japan, which is a riser drilling vessel, which allows for deeper drilling, which is really useful for drilling the Nankai Trough off of the coast of Japan. And then we also, of course, we have mission-specific platforms where we have environments where the water may be too shallow for conventional drilling, or environments which are rather inhospitable, such as the pack ice of the high Arctic. Here's the ASEX cruise, where there are 2 icebreakers which are breaking up the ice to allow for a custom drilling vessel to drill within the high Arctic. And then here's one of the shallow water platforms. This is a jack-up platform from the Chicxulub expedition 364, which drilled the impact crater, which caused the death of the dinosaurs. And of course, there are dozens, if not hundreds, of vessels, such as the UNOLS fleet, which operate shallow coring, such as gravity, piston coring, box coring, etc., to extract top sediments. So once we have all of our sediments up onto the boat or on shore, we can sieve them and clean them in all sorts of ways and extract our nice little bugs coming out of there. And luckily, a lot of the bugs are stored in a number of these fantastic databases and these fantastic archives, which I recommend probing to look at if you're looking at some archived records. So this is how we began. We got all of our previous archives and extracted all of the planktonic foraminiferal data to begin creating Triton. And from there, we identified all of the scientific ocean drilling sites that were missing from the collection of databases, performed a literature mine to make sure our taxonomy was up to date, all of our ecologies, and our chronostratigraphic data was up to date. So biostratigraphic events, polarity reversals, and isotope stratigraphy, which would be used to create new age models for our new sites. And using these chronostratigraphic tie points, we use that to model sedimentation rates for every site and every hole. So it would give every single foraminiferal occurrence an exact, well, a relative date on when that foram was deposited. And of course, if we have the relative date of when the foram was deposited, and we have the coordinates of where the site was initially drilled, we can use plate reconstruction models such as the Seaton model, the Matthews model, and reconstruct where that foram was deposited in time. So eventually this leaves us with up-to-date taxonomies, ecologies, ages of deposition, and the paleo coordinate at the time of deposition of each single planktonic foraminifera. And if we look at our results, if we compare the planktonic foraminiferal aspect or part of Neptune to Triton, Triton now contains 4.66 times more occurrences than Neptune's for planktonic foraminifera anyway. And to put that in perspective, there's over 500,000 occurrences of planktonic foraminifera now in the Cenozoic. And if we compare that to the PBDB, the most well-represented group in the PBDB is the bivalvia, and that's for the entirety of the Phanerozoic. We have just under 200,000 occurrences. That may have been updated now, actually, so I think it's probably more over 200,000. But this is a very, very large database, which contains a lot of records just for the Cenozoic. We quickly look at spatial coverage. You can see that Triton now fills in a lot of the oceans, and you can pretty much see all the continents now, the outlines of the continents. And this is primarily due to the adding in those core top samples from such things as the UNOLS fleet vessels. And if we look spatio-temporally as well, on the y-axis we have our latitudes, and on the x-axis we have our ages from left to right is going older. As we can see, the temporal coverage is really, really good in Triton, especially in the deeper parts of the record. In the mid-latitudes, however, in the lower latitudes from around 40 million years and backwards, it's still very gappy, which requires some more work to resolve these low latitudes and what kind of data we can actually extract from them. So to summarize, Triton now represents the largest occurrence database for any one fossil group, and Triton will continue the amazing legacy of previous data sets and help address key questions within our science. So now we move on to the main part of the talk. After introducing Triton, we now look at the application of networks to the Triton dataset and to answer questions on Cenozoic planktonic foraminifera, how networks can help us understand how different aspects of planktonic foraminifera biogeographically are responding as a community via different connections, which can be elucidated via networks. So what is a network? It's a very good place to start. So a network will be made up of can be made up of any data set, any database, and let's say it's the Triton dataset. So first of all, what we have here is called a node. And a node can be any part of a data set. It can be a species name, it can be a paleo coordinate, it can be an ecogroup or a morphogroup. And of course, within a data set, there'll be more than one of these nodes. So these nodes can be anything. And between these nodes, there will be different connections and associations. And these within network science are called edges. These lines are called edges between nodes. And then, of course, data sets are going to have a lot more than just two. So we can add a lot more of our nodes in, which have all of their own separate edges and associations and connections. And we can use the width of these edges to determine, let's say, what's the frequency of associations or the strength of associations between different nodes. And now that we have multiple nodes with multiple connections, this entity is now what we would call a network. And networks operate in many different classes depending on what kind of nodes you have. So you can have multiple different node classes. Let's say in this network we have ecogroups, morphogroups, paleo coordinates, and also species occurrences. But there's a special class of networks which only uses two distinct node classes. So when you only use two node classes, let's say ecogroup and the paleo coordinate, what we have then is called a bipartite network. And we can arrange these bipartite networks within their respective nodes to look at the relationships and the associations between these different node classes. And these bipartite networks have recently been successfully applied to a lot of modern ecology and have now been started to be implemented into paleobiology. So here's a study from my colleague, Dr. Anshuman Swain, from 2021 in the journal Paleobiology, using bipartite networks to look at the association between damage types from insects on leaves and leaf species. So the green bars we have here on the bottom are our species of leaves. This is one of our node classes. And on the top here, we have our damage types, which correspond to our key here, the functional feeding groups of leaf damage, such as galling, overposition, margin feeding, leaf mining, and things like that. So this is a snapshot in time where we have different species of leaves and how different damage types are interacting with them. And Anschumann and others showed the evolution of how these different damage types have changed through the fossil record. But of course, instead of damage types and leaves, our different node classes, as I mentioned, can be, say, ecogroups and paleolatitude, or morphogroups and paleolatitude. So the methodology for our network analysis was to take all of Triton and bin the entire data set into paleolatitudinal bins of 5 degrees. And then all data was also binned into 1 million year time bins for the last 66 million years. So 66 time bins, and also at a higher resolution for the last 15 million years, we binned our data into 250,000 year time bins. So what do our Cenozoic, Planktonic, foraminiferal bipartite networks now look like? So here is the bipartite network for the last 250,000 years. And on the right, we have our ecogroups. And on the left, we have our 5-degree paleolatitudinal bins. And let's add a little more information to that to make it a little clearer what's going on here. So in our ecogroups, we have the main 5 ones. We have our surface mixed layer species, thermocline, subthermocline, and high latitude. We're missing our high productivity forms, our green ones up here, because they only exist during certain periods of time through the Cenozoic. And they don't exist currently in the last 250,000 years. And on the left here, we have our paleolatitudinal bins, each of 5 degrees. And I've put in the equator here to give us a little bit more information of where we are. And we have our different colors here ranging from red to blue. And what those different colors mean, it's a index that we've based on on previous indices within these studies, which we call these colors will also be consistent throughout the rest of the figures, where we have either the ecogroup specialization index or the morphogroup specialization index. And what this index measures is how equitable the distribution of ecogroups is in a given latitudinal bin, these 5-degree bins here, for a given time period, in this case, the last 250,000 years, where red values show low specialization. So low specialization meaning if we have a value of 0, close to 0 like it is here, that would mean that all of the ecogroups present within this time bin are present in pretty much equal abundances, equal occurrences, equal distributions. So everything in that time bin, everything is getting on very well. Whereas if we have a blue value is high specialization, which as we can see down here, this is a very clear blue one here in the southern high latitudes. If we can just trace this gray line back here, it's being dominated by our high latitude forms, of course. So Neogloba Quadrina pachyderma is the only thing in that bin, and it's having a really good time on its own. So this is a single bipartite network for the last 250,000 years. So within our study, what we've done is we've constructed individual bipartite networks for the last 66 million years and also the last 15 million years. So here we have the last 15 million years, an individual bipartite network stacked along horizontally. Time is on the bottom of these plots, and on the left here we have our paleolatitudes. So these are all individual bipartite networks stacked along, and this is for the ecogroup specialization index. So this is for the last 15 million years, which I'll detail what's going on in just a second. First of all, we're going to look at our bipartite networks for Cenozoic morphogroups, so the 19 morphogroups. And this is for 66 million years here, again, paleolatitude along the side. And here are the oxygen and carbon isotope data from Thomas Westerhold. So we have our, moving from the right to left, we have our intensification of northern hemisphere glaciation around 3 million years ago, our Miocene climatic optimum from 17 to 15 million years ago, a Oligocene-Miocene boundary around 23 million years ago, Eocene-Oligocene boundary around 34 million years ago, the MECO, Middle Eocene Climatic Optimum around 40 million years ago, the early Eocene Climatic Optimum around 50 to 54 million years ago, and the Paleocene-Eocene thermal maximum around 56 million years ago, and of course the Cretaceous-Palaeogene mass extinction, which started the Cenozoic. So just looking at this very briefly, we can already see very clear patterns sticking out. First off, immediately following the Cretaceous-Palaeogene mass extinction, we appear to have generally high specialization across all latitudes, which is likely due to the fact that pretty much all planktonic foraminiferal species were annihilated across that boundary, and we only have our Miraco headbagelids within the macro perforate species surviving. So it took a while for species to regain that morphological diversity, as shown by Chris Lowry and Andy Frass. And then also it appears that we, in the southern hemisphere specifically, it's very high specialization, which may be due to the fact that following the KPG extinction, Brian Huber et al. Showed that there appears to be very high endemism within the southern high latitudes following the mass extinction. Moving forward, one thing which is very clear is that we appear to have a very big signal of what biostratigraphers would call the Uglyocene, when unfortunately all of our beautiful species, beautiful ornamented species such as Hancanina were wiped out across the Eocene boundary, leaving behind our globular forms such as Dentoglobigerina, Sabatina, and Paraglobiritalia, which dominate the record for most of the Oligocene. So as we can see here, it looks like across the Eocene Oligocene boundary, someone smeared a blue paintbrush there, dropping from low specialization in morphogroups before and after the Oligocene to high specialization, where we have mainly the same morphogroups across all latitudes. And interestingly, it appears that even prior to the Eocene-Oligocene boundary, we have in the southern hemisphere, especially our blue high specialization form signal is already encroaching upon the Oligocene. So what I believe this is due to is following the descent into the ice house after the MECO, we have more and more Antarctic water influence, which is pushing species equatorward, where we have dominance of cold-loving species within the high latitudes. Not so much in the northern hemisphere, but definitely within the southern hemisphere. There's a very strong signal for this. And then following that, around 19 million years ago, there's a very abrupt reduction in specialization. We now switch from blue to red, which may be further evidence for the hypothesis put forward by Elizabeth Seibert that sharks nearly disappeared around 19 million years ago. So this abrupt change around 19 million years ago may actually indicate a change in the marine trophic resource continuum at this time, giving more evidence to this extinction within pelagic sharks. So now moving on to the ecogroups, our 6 ecogroups indicated up here. Again, this is again the Cenozoic 66.0 and the paleolatitudes here, and it's the ESI, the Ecogroup Specialization Index this time. And again, patterns start to pop out just at a first glance. So obviously here during our early Eocene Climatic Optimum, we have very high specialization in blue, indicating very few ecogroup species dominating these latitudes here, which may be due to the fact that across the Paleocene-Eocene thermal maximum, we have bountiful evidence of not only temperatures such as 40 degrees centigrade in tropical waters, which are prohibitive to marine life, but also a lot of migration away from these low latitudes during the PETM. And maybe that it appears that maybe this wasn't just a PETM signal, that maybe that this migration away from low latitudes was also a early Eocene climatic optimum signal. However, there's also, of course, there's a huge gap in the lower latitudes, so it's actually currently not possible to give accurate latitudinal biodiversity gradients during the hothouse world of the early Cenozoic within the marine plankton. Also, interestingly enough, the southern hemisphere once again appears to potentially act like a refugia, where we tend to have more generalized occurrences, lower specialization. And seems to be that more things are getting along well, and all occupying the same region. And this may be due to the fact that we had a pronounced zonal heterogeneity longitudinally within the southern hemisphere. So there was lots of different niches and environments that may have acted as refugia for the planktonic foraminifera at this time. And then moving forward in time, as we push forward into the last 5 million years or so, we can see there's very high specialization within the high latitudes. And this is mainly due to the fact that, of course, we have neoglobal quadrina pachyderma and other high latitude forms completely dominating our polar assemblages and faunas up in the high Arctic and Antarctic. And then most interestingly, however, in the last 8 million years or so, we appear to have this impingement of zones of low specialization, like zones of generalization, which appear to be migrating towards the equator over the last 10 million years or so. So I'm going to detail what we think may be going on here. So this is that same time period. This is a zoom in of the last 50 million years, though. And again, paleo latitudes on the side, they're slightly different colors and of the Westerhold carbon and oxygen here. So here's the mid-Pliocene warm period and the descent into the bipolar cryosphere here. And what we think is going on here, we see our pincer movement of our zones of red as we move towards the modern. And I think that this may be due to associated with the fantastic work done by Flavia Boscolo-Galazzo and others, showing that around 15 million years ago, planktonic foraminifera and calcareous nanofossils started to pioneer and migrate down through the water column into the twilight zone of the ocean, originating within the mid-latitudes. And they were following food and water, food and oxygen down into deeper depths. And so we've made this schematic to indicate what may have been going on for the last 8 million years or so after that initial migration down into the water column around 15 million years ago, where our data is a little more spotty here. So it appears that around 8 million years ago, following the work of Flavia, we have this oxygen and food moving down in the water column, and our forams will be following that food and expanding their ecological niches, and then moving upwards through time from 4 million years ago to recent. Here's a schematic of what's going on. Those blue bands are actually the red bands over here. We have oxygen and food moving not only deeper, but also equatorward. So our deep dwelling species will actually be following that food from the mid latitudes down into the lower latitudes. So they're tracking that. But if our red colors indicate that all of our ecogroups are getting along very well and occupying the same water masses, then obviously the shallow dwelling species also need to be migrating equatorward. So what we believe is happening is that as the bipolar cryosphere is intensifying, we have more and more pack ice, more and more cold water is impinging across surface waters in the high latitudes, intensifying our bipolar cryosphere. And as that cold water is impinging equatorward, we have not only migration of our deep dwellers equatorward, but also of our surface dwellers. So it appears that the entire clade has shifted its ecological communities towards the equator by around 30 degrees over the last 8 million years or so. So importantly, what these red zones represent, these zones of low specialization, zones of generalization, equitable zones, is that they do not reflect zones where populations are most concentrated, nor necessarily where species are at their ecological optima. Instead, what they do represent is global ecotones. And these are regions where latitudinal conditions are the most equally accommodating to all of our ecogroup and morphogroup members during each of our temporal bins. So the morphogroup, this was the ecogroups I was showing you, but the morphogroups do show a very similar pattern as well. So how do this pattern, this latitudinal pattern, how does it relate to the latitudinal biodiversity gradient? Well, we take our zones of low specialization that I just illustrated, and we compare them to our modern latitudinal biodiversity gradient amongst the planktonic foraminifera. And if we compare the two, we actually get a very, very strong, significant correlation. So we were thinking, oh, maybe that this pattern consistently is the same as the latitudinal biodiversity gradient through deep time. But actually, if we compare and correlate the latitudinal biodiversity gradient versus our ESI patterns, it's the strongest we get is actually for the last 250,000 years. So along here, we have the last 15 million years. And along the side here is the correlation between these two patterns. And red dots indicate where we have statistical significance. So actually the strongest relationship is in the last 250,000 years. But as we move backwards through time, this relationship quickly breaks down, it disentangles, and around 4 million years ago, it actually did not exist. And it appears to be random on what's going on within deep time. So it appears that what the pattern we're recording within our ecogroups and our morphogroups is something that's entirely separate from the latitudinal biodiversity gradient. And what we've been calling this is the latitudinal equitability gradient. And this is the gradient of community equitability recorded across functional groups such as morphogroups and ecogroups in planktonic foraminifera. And why it's important to be using functional groups, or you could use functional traits, functional characters, or just any characters. It doesn't have to be, doesn't have to have a known function, is that these traits and groups can be consistent across an entire clade. So our entire Cenozoic phylogeny can be broken down into 6 ecogroups and 19 morphogroups. And where species are evolving and becoming extinct, as we move through time, we have some 350, 360 species of planktonic foraminifera. At the same time, these ecogroups and morphogroups can stay the same. So we can look at the entire phylogeny across large swathes of time and not have to account for evolution or extinction within the group. And we hypothesized that these LEGs, or latitudinal equitability gradients, likely exist across multiple clades and will be of different shapes depending on what that clade is doing and where that clade prefers to live. But why is this new gradient important? Why are these legs important? And that's because in the modern ocean, where our LBGs and our LEGs are very highly correlated, the biodiversity hotspots and the biodiversity gradient of planktonic foraminifera matches very well the gradients of macrotrophic resources such as tuna, billfish, krill, and squid, all resources which are very important to billions of people around the planet. And the key question is, considering that this relationship between LBGs and LEGs breaks down in worlds in 4 to 5 million years ago, when CO2 levels are equatable to how they are in the modern day, are our modern motile predators, which are key trophic resources for humanity, are their distributions similar to planktonic foraminiferal LBGs, or are they similar to LEGs? And of course, that's a problem. As I mentioned, our CO2 levels are now high, 415.64 parts per million as of yesterday. And of course, this 415 parts per million is higher than even our most conservative estimates of the Miocene, and currently impinging upon Pliocene levels, which have not been seen for 3 to 4 million years. So 3 to 4 million years ago, or even 8 million years ago, when our LEG low specialization spots were around 60 degrees north and south, if we have CO2 levels which are now equatable to those, are we going to be seeing resources and organisms moving towards 60 degrees north and south of the equator and staying there, which is, of course, it's difficult to ascertain and calculate exactly what would happen if these organisms, which are evolved to live within lower latitudes, are now living in higher latitudes, and this could break down entire trophic levels of the ocean. And even more interesting, actually, is that if 8-ish million years ago, we have our LEG low specialization regions where our ecotones, where everything's getting along well, at 60 degrees north and south, that's quite close to the Arctic Circle. So if we look at the last 66 million years, how were our communities operating, and what would our networks show us if the resolution was as good as it is for the last 8 million years, if it's was as good for the last 66 million years? And how do our networks operate in our greenhouse worlds of the past if we can get them to a higher resolution, our greenhouse and hothouse worlds of the past? So to summarize, networks are clearly an incredibly powerful tool in paleobiology and for looking at the planktonic foraminiferal record. And our morpho groups showed very high specialization post KPG and potential endemism within the southern high latitudes. And of course, that ugly Eocene really picks out very nicely, showing high specialization, again, within the southern hemisphere, especially, and across all latitudes, we have higher specialization of morpho groups. And potentially further evidence of the early Miocene shark extinction put forward by Elizabeth Seibert. Within our ecogroups, we showed that they were highly specialized during the earliest Eocene climatic optimum, possibly due to the thermally prohibitive temperatures within lower latitudes, and it appears potentially there may have been a southern hemisphere refugia amongst the Cenozoic planktonic foraminifera at this time. And of course, most importantly, we have these low specialization zones moving equatorward for the last 8 million years, which caused us to coin this term, the latitudinal equitability gradient. And we need to work out how that relates to the biodiversity gradients of other marine groups. So I'd just like to acknowledge a lot of people, too many to name individually. And of course, I'd especially like to thank the Cushman Foundation for Foraminiferal Research and the Micropaleontological Society for asking me to give this seminar, and also for all of you there for listening to me talk about forams and to talk about networks. So thank you very much for listening.
Oh, that was great, Adam. Thank you. So we have time for some questions now, if anybody has any. We do already have one in the chat from Lizette. So she asks, the abundance of pixels increases as we have younger records. Is that because of the density / availability of data sets? So for example, it seems lower in the higher latitudes in the Paleogene. Is that correct?
That is 100% correct. For the last 15 million years, we tried a number of different resolutions of pixels from 1 million years to 100,000 years. And actually, for the last 15 million years, our pattern in our low specialization zones and our LEGs, that showed up whatever resolution we tried out. So that pattern is consistent across our different resolutions, but 250 kiloyears appeared to not show it best, but it was the one that we settled on in the end. But the reason basically is that as you can see in the southern hemisphere especially, we actually have high latitude records for the last 66 million years only for the last 5 million years. And this is mainly due to the fact that coring in those southern high latitudes is very difficult due to the ice rafted debris. And there's just so much sediment that comes off of Antarctica that you just cannot get past the Pliocene usually. And of course, forams as well do not preserve well in the high latitudes. So the different pixel size, the different resolutions is strictly due to the fact that the record is just so much better for the last 15 million years, and especially the last 8 million years, because you can see it does get a little spotty for the last beyond 8 million years ago.
Yeah, that makes sense. So we have another question from Katia in the chat. With the heating of oceans, what has been observed? Sorry, let me start that again. With the heating of the oceans that has been observed in the migration of macrofauna, so predators, to higher latitudes, contrary to what you showed for foraminifera at PETM, for example, how could this happen?
How could them moving to high latitudes happen in the modern or in the PETM?
Is Katia still here? Did you want to clarify your question? Not sure. Just trying to understand. So if Katia wants to clarify that, maybe if she wants to put in the chat. But if anyone else has any questions, you can raise your hand and speak, or you can put them in the chat.
I think I understand the question actually. It does actually show in the PETM that forams do appear to have migrated towards the high latitudes during the PETM, but specifically the southern high latitudes, which is basically exactly what we're seeing today. From the paper from Lucas Yonkers 2019 actually showed that planktonic foraminifera have shifted away, polewards away from from their pre-industrial biogeographies, and they're actually actively, not actively because obviously they are mobile, not motile, but they are moving away from the equator as the Earth is warming. So we're seeing this, the exact same thing. According to our networks, we're seeing the same thing in the PETM as we're seeing in the modern day.
Thank you. Does anyone else have any other questions? I can't see any hands up or anything in the chat, so. Oh,
well, anyone can send me an email if they'd like to ask me any other questions. Okay.
How can your results be applied to a future climate change scenario? I think that's quite a good one.
Yeah, very good question. I guess what we could do is we could take strict time points for the last, say, 8 million years or so, where we have good data and we have good data of what CO2 levels might be like. And we can potentially model where, if we just created a model climate scenario of CO2, say 500 parts per million, and we took our modern fauna and we looked at maybe their pre-industrial distributions, and we put them into the model and found out where they went, we could actually produce the networks moving forward in time for different climate scenarios.
Yeah, that's good. And then we have another question. So we have, is the ESI a proxy of functional diversity, not species diversity?
It is somewhat. So our functional diversity does show slightly similar patterns, but it's only if you would use functional Shannon diversity, you do get very similar patterns to our ESI and our morphogroup specialization index. So it's a good question, and yeah, we do get similar results to functional diversity as well.
Okay, brilliant. Are there any other questions? Marina, did you want to unmute? You can ask.
Yes, hello, I'm gonna unmute. Sorry that I cannot put my video on. Adam, thank you for the talk, it was a great talk. I have a question, I think also regarding the specialization index. I was wondering if you did some, or if there is an effect of the amount of data that you have for each of these grid cells. For example, if there was a sensitivity analysis, maybe subsampling the data to have, because I can imagine that some of these squares sometimes have just one data set, and some of them have many, for example, and that can maybe influence how you get, how this index is calculated. I don't know. So I was just wondering if there was a sort of sensitivity analysis regarding that.
Yes, so in the supplementary data for our paper that's in review at the moment, we did actually perform null model analysis to look at whether or not the patterns, we compared null models to the patterns we're seeing, and where we're seeing our important patterns within the last 8 million years. The null models showed that these patterns are different from what we'd randomly expect if we just took into account the data as a whole. So we have performed that analysis as well.
Very nice, thank you. Looking forward to their paper.
Oh, thank you. Yeah, me too.
That'd be great. So I think that's all the questions. Adam, did you say if people have any more, they can email you?
Yeah, they can.
Okay, brilliant. Yeah, if
they can.
Something, send an email to Adam. And thanks again, Adam, for your talk. And thank you everyone for attending. We don't currently have a speaker lined up for next month, but we will have one. So as soon as we have any information, we'll send that all out for you. So yeah, have a good weekend. Thanks, everyone.
Thank you, everyone. Bye-bye.